onto the chat room chat, and then we'll review this question after the presentation. All right. Okay. Now again, back to you.
All right. Cool.
And also, I guess you just see something pop up to say that the meeting is being recorded. So I hope everyone is fine with that.
Yep. So everyone can see my screen now. So my presentation today is structured into 3 parts: who we are and what are our aims and what we have done so far to achieve our aims. And I will highlight it only in 3 areas today. So for some of you are very familiar with us, feel free to switch off and make a coffee for yourself. But lastly, I would like to offer some opinions on how the future will look like, in particular how the COVID-19 will shape the dairy industry. So who we are, our full name is Australian-China Joint Research Center in Future Dairy Manufacturing. Our center at MONASH is funded by federal government from May 2016 to July this year. As you can see from this structure, joint forces with the other 3 affiliations in Australia and China. 2 of them are the GOA University and 5 industry partners, including 4 leading companies. 4 leading dairy companies: Saputo, Fonterra, Mengniu Dairy, Bega Cheese, and also the Corrugation. The center is clearly an industrial point, providing Australian company with access to local and international expertise, market research, and also the innovation through the high quality research. In Australia, we have 5 scientists, 3 postdoc researchers, and 10 PhD students working at 4 research themes. And today, some of our proud PhD students will present their up-to-date research outcome. And our aim, in summarize, our aim is to enhance the relationship between dairy and researchers industry in Australia and China. It's to establish or strengthen the research and industry partnership and collaborations. And lastly, foster skills and talent for dairy innovation, as well as attract, train, retain skilled professionals. In order to achieve those aims, firstly, we conducted intensive exchange visit to China and also host visit in Australia. For example, in 2018, we organized the 2nd joint symposium focusing on cheese technology. On top of the technical presentation, similar like what we're having today, we invited cheese experts from Iran, France, and also the industry speakers from leading companies. In particular, we organized a special cheese wine exhibition where the Bega, Fonterra, Australia, and Mengniu, our partner, and also the new partner, Ala Foods, exhibit their signature cheese products there. We had a good time there, maybe partially because of the wine as well. We also took the opportunity to know more about our partner by visiting Corrugation NHRI and 3 Mengniu manufacturing sites. November last year, after our 3rd symposium in Suzhou, we visited some leading companies such as DuPont, Cargill, Mondelez, Janus University, Suzhou University, and MONASH Suzhou campus. We were lucky because the trip was happening in November last year. We managed to go back safely, no problem. And of course, the visit is always two-way street. Early last year, we hosted the Vice President of Mengniu, Dr. Wei Zhuyu, and also the R&D Director to visit MONASH, where we had some in-depth conversations on how the Mengniu and MONASH can work together to innovate in the membrane, food packaging, and innovation training. Secondly, to link research and industry, until today, as Mark mentioned, we have organized 17 ICGRC exclusive workshops and presentations, engaged with over 700 industry participants. And it's worth to mention that all these workshops are being recorded to benefit those who could not join in person. So if you have missed any of them, feel free to reach out to me for those recordings. And lastly, and most importantly, we achieve our aim via the innovative and industry-based research. under the Ford research themes, developing dairy function products, innovative membrane processes, modeling for process optimization. We attract, train, and retain skilled professionals, result in 3 students submitting their thesis, and 1 is currently working in HPS engineering, and 1 PhD now become a postdoc. And also the postdoc team, that 1 postdoc become a lecturer in food and 1 postdoc become a senior postdoc in New South Wales. So we are proud to what we achieved so far, and it wouldn't be possible without all of your support. While looking in the future, we also continue to hope to continue collaborating with you, as Mark and Cordia mentioned earlier. And on the other hand, when we look into the future, inevitably the impact of the COVID-19 will carry on. Personally, COVID-19 affecting my life significantly, because my parents are in Australia for 4 months now, and potentially they also need to extend for another few months because there's no flight back to China. And it is hard for me, and I think it's even harder for my husband. So we actually agree that our marriage is our priority. So yeah, it's something about my life. And I think how the COVID-19 affecting the dairy industry in China might provide some useful insight for the industry in Australia as well. So I would like to take you through some data here. So first of all, let's look at the Chinese dairy consumption in 2019. In this table, you can see that in 2019, the annual per capita of dairy consumption in China is very low, about 35 kilo, which is equivalent to less than 100 grams per day. And the main product is drinking milk and yogurt. In contrast, Australian consumed 125 kilo with intensive variety like milk, cheese, butter, and yogurt. However, the market size of China is huge despite the very low consumption per person. In this figure, you can see that the size of the bubble indicates the retail value of dairy product, and China is listed as a second place globally with $61.7 billion in 2018. The China market is huge, and especially it relies on the imported dairy products. According to the General Administration of Customs of China, 2.97 million tons of dairy product, which equivalent to 16.6 million tons of fresh milk imported to China, and increased by 12.8% as compared to 2018. And those imported goods are mainly from New Zealand, of 40%, and followed by Germany of 12% and Australia 7%. On the other hand, Australia actually sits in an advantageous position. According to the free trade agreement between Australia and China, the tariff for yogurt, butter, cheese will drop to 0 in 2024. And by 2026, all the Australian dairy products will charge to 0 on tariff when exported to China. And let's take another look at the production of the dairy goods during the pandemic. There's some data from January to February 2020. Because of the COVID-19, the production of the dairy goods decreased dramatically. As you can see that liquid milk dropped by 70.5% year over year, cheese by 60%, and milk powder by 15%. The reason is the manufacturer ceased the production during the Chinese New Year, and also unfortunately during the pandemic period. And also the close of the offline retail and food services, which is seen in the cases here, which leading to the excessive stock up. The good news is, as an essential business, dairy industry is one of those industries which have early approval to resume production. In March, over 685 are back to business now. However, the demand of the dairy consumption is actually increased in China because of the pandemic. We got to admit that the pandemic has fundamentally changed Chinese consumers' behaviors on how they are reaching to brands and products and where they're buying and also what they are buying. The dairy consumption increased during the pandemic and also expected to grow post the pandemic because of the following reasons. First of all, the endorsement of the nutrients of dairy and their contribution to boost the immune system. It's acknowledged through the official channel. February 2020, the National Health Commission of China published a set of guidelines of dairy consumption for local residents in a hope to strengthen the public immune health to fight the outbreak. And in the report that the milk is an excellent source of high quality protein, and it is advised for a daily intake of 300 grams. We mentioned earlier the annual per capita of Chinese is 35 kilo, which is less than 100 grams per day. And in this guideline, the recommendation daily intake is 3 times higher. Also, the high demand were also contributed by the celebrity endorsement from the senior public health advisor, Dr. Zhang Wenhong. His influence in China is equivalent to Dr. Anthony Fauci in the United States. During an interview, he strongly recommended milk as an essential part in breakfast during pandemic and post-pandemic. And secondly, the online channel has become the most effective selling channel, and many companies have their own e-commerce channel prior to the pandemic. In Alibaba, Q1 2020 data shows that the sales revenue of infant formula increased 38% by 38%, and Atamil and A2 are the top 1 and top 2 respectively. Lastly, change of the lifestyle. Stay-at-home mode triggered the DIY lifestyle, and the homemade dishes become ever popular in China. There's a snapshot of the Tmall sales on a certain day in March 2020, shows that, of course, the hand sanitizer wipes increased by 422%, and grocery for foods increased 180 to 290%. Here, bakery ingredient actually increased by 140%, followed by the personal skincare. So following this trend, I personally believe that the following products have the potential to grow post-pandemic. That is the formulated powders, such as lactoferrin added powders and yogurts, as it recommended as a daily intake for the mild infective COVID-19 patients. And also the probiotics related to immune system and the cheese buttercream related to the homemade dishes. Lastly, I would like to share with you the Chinese word of crisis. Weiji, the first character Wei means danger, and the second character Ji means changing point. It means that the risk and opportunity is coexisting. In this particular time, I hope that we can turn the lemon into lemonade and turn the crisis into opportunities. And at the end, thank you for your attention and happy to get the questions. Oh, I can.
Yeah, sorry. Hi, it's Jayshree here from Bega Cheese. How are you?
Thank you, Jeffrey. Yeah. Thank you.
Lynn, thank you very much. A very interesting presentation, and also just really good overview of, good to see what the consumptions are and so on. I was just, being a bionutrients technologist, I was just interested in your comment about, in your opinion, you thought the formulated milk products with lactoferrin. Can you maybe elaborate a bit on that? Has there been any interest, or has there been any interest or any products out there that's kind of indicated that, please?
Yes, thank you for the question. Actually, when I think it through, I think the formulated powder is not just refers to the infant formula, because that during the COVID-19, the vulnerable group of the people is actually the aging people. People will worry about their parents or their senior family members will get affected. First of all, they protect them, and secondly, they will also willing to purchase products which has the function to boost their immune system. And lactoferrin has been well known in China so far. Gradually getting attention in terms in his contribution to the immune system. So I think it's not just the infant; it's also related to the specially designed for a special designer for the certain group.
Yep, thank you.
Is there any questions from the audience? Any other questions? Or perhaps if there's any question that comes to mind, maybe you can even write it in the group chat, okay? And we can certainly follow up on the questions along the way. No questions at all. In fact, when I hear Lynn talk about the increase in baking, 140%, that really resounds a lot, because I think especially the DIY, everyone is doing everything at home. Yeah. Mm-hmm.
Yeah, I think I can comment a little bit on that. So unlike in Australia, we are allowed to go out for exercise or visiting essential families. In China, actually, the complete lockdown is that you can only go out for the grocery shopping. So there's not much you can do. And also most people living in the apartment rather than the houses. So there's not much activity you can do in terms of DIY. So cooking is one of their favorite work to do. If you have some Chinese friends, you've probably seen them actually showing their cooking skills getting improved during the pandemic. Yeah, of course, their face is getting a bit big as well, because they gain some weight as well. Yeah.
All right. If there's no more question, I think I'll invite the next speaker, Dr. Evan Hein. Evan, if you can share your screen with us.
Yep.
Okay.
Wait a minute.
Yeah.
Share my screen.
Okay. Now, just allow me to give an introduction of Evan. Yeah, Dr. Evan Hein is currently an acting assistant manager in the International Science and Innovation branch in the Department of Industry, Science, Energy and Resources. He began this role in the second half of 2019. He originally joined the department in late 2016 in the Timor-Sea section in the offshore resource branch. So he also completed his PhD in political, international, and strategic studies at the ANU in 2017. And on that note, Evan, over to you.
Hello, good morning. Thank you for the introduction. Just as background, in my role and what our branch does, we play, in addition to playing an important part of the Australian government's response to the COVID-19 pandemic, one of the department's broader concerns is to support science and its commercialization. My branch is specifically concerned with the thank you to the Symposium and for hosting this, particularly through teleconference and Zoom. It's great to see. Hello?
Sorry, Evan, you got a bit choppy.
Yep.
May I suggest maybe we can try turning off the video and see if
Sure.
The connection improves. Yeah. Okay. You
Stop my video.
at the bottom left there is. Oh, I can turn it off. Oh,
Is that
okay? Yep. Oh.
okay? All right. So okay. Hopefully this works. Where was I? All right. So today I'll be speaking briefly about the Australia-China Joint Research Centres and how they can advance innovation and technology, including a quick look at how this has played out with the future dairy manufacturing as a case study. So in this presentation, I'll give a quick overview of the Australia-China Science and Research Fund, the ACSRF, with a particular focus on the joint research centres. And then I'll be taking a look at the future dairy manufacturing and how it demonstrates how terrific and commercially successful advances in innovation and technology can come about from Australia-China science collaboration. So slide 3. The science and research relationship between Australia and China is well developed, having began before formal diplomatic relations. In 1980, of particular concern, the Australian government entered into a treaty-level agreement with China on science and technology cooperation, and this year marks the 40th anniversary of that. The treaty underpins the Australia-China Science and Research Fund, which my department manages from the Australian side with our Chinese counterpart, the Ministry of Science and Technology. The Australia-China Science and Research Fund, or ACSRF, began in 2011 with its goal being to support strategic science, technology, and innovation collaboration of mutual benefit to Australia and China. The program's intended outcomes are to bring together leading institutions in Australia in a particular research field with Chinese partners to achieve economic, environmental, and social benefits for Australia and China, maximize the application and commercialization of research outcomes, and provide early career researchers the opportunity to gain relevant Australia-China research experience. The ACSRF has 3 elements: thematic workshops, seminars, and symposia that seek to strengthen cooperative research links between Australia and China in areas of mutual interest. The 15th symposium held last year in Melbourne was on precision medicine. The second component is the Young Scientists Exchange Program, or YSEP. This provides an opportunity for early to mid-career researchers from Australia and China to undertake a 2-week exchange. So far, 277 Australian and Chinese researchers in a range of fields have participated. Past participants have actually gone on to successfully apply to be project leads for the joint research centres, which include famously Professor Selamulia, who participated in 2013, and she spoke a bit earlier. She went on to great fame as director of the Joint Research Centre in Future Dairy Manufacturing. Of particular interest today, of course, though, is the Joint Research Centres. This is a very important component of the ACSRF. They are virtual centres that link Australian and Chinese research institutions conducting research in priority areas agreed to by the Australian and Chinese governments. The aim of the joint research centres is to build capacity, research capacity, maximize the application and commercialization of research outcomes, and boost researchers and industry. Since 2011, 17 joint research projects across 3 rounds have been supported across various priority areas. These have included energy, environmental science, food and agriculture, its manufacturing, and medical technology and pharmaceuticals. These priority areas are discussed between the governments before each round opens for applications. So how do they work? They are available to Australian organizations doing a research project with China in a priority area. Australian research organizations need a Chinese partner who applies through the corresponding Chinese system. For the Australian side of things, there's $1 million over 3 years, which is then matched from the Chinese side for the Chinese partner. And when you enter into the grant agreement, the department manages the grant by working with you, monitoring progress and making payments. Where we are now given the COVID-19 situation, we have prepared a round 4, but we're just holding off on launching it for the moment until things become clearer about the future of travel and research. The department also has various support networks available to aid of engaging with overseas organizations and governments. The department has counsellors located in Beijing that engage with industries and science and research institutions to encourage collaboration through Australian government initiatives. Our counsellors are also engaged with foreign governments and support Australian industry development, innovation, and expansion overseas in key growth sectors. Esther Harvey is the current counsellor dedicated to science and technology. Scott Wilson is the counsellor for resources and energy, and their details are available on the slide in front of you. There's also a counsellor dedicated to intellectual property from IP Australia, David Bennett. You can also scan the QR code there if you prefer it, and that will link you to the Chinese Embassy's website, the Australian Embassy in China's website. Intellectual Property Australia also has published education materials and guides to assist Australian researchers and businesses in approaching global markets and protecting intellectual properties overseas. Lastly, the Department of Education, Skills and Employment has developed the University Foreign Interference Task Force Guidelines. These guidelines were created to aid the university sector in protecting themselves against foreign interference through supporting an environment of trust and guiding decision-making based on potential risks. They've done this to provide Australian universities can continue to produce world-class research. These guidelines can be found on the website of the Department of Education, Skills and Employment. As Lynn has already gone into in great detail on JRCs, I don't want to look at the future dairy manufacturing too much, but I do want to say it is a great success story, and the department's very, very proud. As you've been informed, it's a partnership between Monash and Soochow University beginning in 2016, which has had flow-on opportunities and for the industry to benefit from two-way knowledge exchange, access to expertise, and access to new and emerging markets for dairy products. The joint research centres aim to provide the dairy industry with manufacturing process efficiencies and new high-value products, which will develop a strong link between Australia and China's large distribution network. Indeed, China is Australia's largest market for dairy exports, for example. It was worth $905 million in 2017, for example. The joint research centres have provided many economic, environmental and social benefits, including greater dairy manufacturing processes to be greater efficiency in those processes and producing quality products that command premium prices. The potential to link Australian dairy manufacturers with the largest supply and distribution chain in China. Improved energy efficiency for energy-intensive unit operations in dairy processing. More efficient cleaning strategies to reduce waste. And finally, the dairy industry attracts, trains, and retains skilled professionals to ensure an agile and innovative workforce, particularly in rural areas. So I missed a slide. But that's all from me, and I'm be happy to take any questions, or if you want to contact me later, I'll leave my contact details.
All right. Okay, thank you, Evan.
I'm sorry for the, I don't mind, my internet connection may not be that great, so I apologize for the.
I think the actually the your voice came out very smoothly from my end. So I think the maybe there's a bit of you know, our individual connection. I think once the video was turned off, yeah. So okay, and thank you for the for your presentation. Now I would like to invite the audience to pose any to ask any question. This is an opportunity, and I think throughout your presentation, the chat room was very alive.
Oh, really? Okay.
Probiotic kind of alive. So the questions may be pertaining to the
comment and also a question as well.
Sure.
I guess, you know, it's, as you mentioned, you know, we have been very fortunate to get this support from the government for the center. And I personally also, and also Meng Wei actually,
Yes.
we were both part of the Young Scientists Exchange in 2013, which actually started the whole, this whole process. And we do appreciate the government support in this. And I know currently it is a challenging time because of all the things that are currently happening in the world. But I do hope that going forward, especially for science and research, China is a very important partners to us and also commercially. And now, of course, as university, we do realize that, you know, Chinese, especially international students from China are very, very important part of our community. And I guess my question is, as you mentioned, there is some delay because we need to understand in terms of the borders and so on. But with regard to this program and also similar program, did you can you comment on what is the position for the government? Will they continue this in the future after everything settled back to normal? And what will be is there any kind of ideas about the shape or the format of the program?
I think the program, are you, sorry, is that, you happy for me to?
Yes, please, thank you.
Yep. I think the department and government has a really positive attitude to the program. I think they're really, I think they see science as a really, science, research, technology is an important sector to invest in and to support, and China's obviously a big partner, and there's a lot of complications around the world with geopolitical issues, but science is seen as an area where a lot of mutual benefits can be produced, and I've only heard positive things, so I think there's a bright future for collaboration. Obviously, there's uncertainty about currently now and what happens after perhaps things sort of go back to some kind of new normal. I mean, things won't go back to the way they were before. COVID has changed the research landscape, so that's an interesting space to watch. But I think they'll be looking to move into that kind of area to really look at what we can do. would you a follow-up question? Does that sort of get to where you're?
No. I think that's kind of, and it is reassuring that it is still a priority, and obviously we have to see where things goes. You know, when the borders will be reopening and all this different stuff. But yeah, it's good to know that it is one of the priority area, and also the area where we can see future collaboration. Thank you.
Yeah, it's very. My sense is it's very positive, and it's an area they really want to take advantage, like push forward. So,
thank you.
And really just echoing what Cordelia mentioned, when we were there, both of us were part of the Young Scientist Exchange Program, and I think the opportunity to be in China to also build up the relationship and just to learn the cultural aspect of how to do research together. Those type of experience is invaluable, actually. Yeah. Any questions?
Yep. Here is a question. This is Chris Su from MONASH University. Thanks, Evan, for your presentation. My question would be, what would be the government's position or support for the post-JRC period? Because we know usually for JRC it has maybe 3 or 4 years for per round, but after this maybe 3 or 4 years, after this period, what would be the government support or perspective?
For future JRC
for future,
round?
Yeah, no, for example, for this future dairy manufacturing, and we all know it will finish this year. So post this funding round, what would be the sort of new or future perspective from the government's position?
For the future dairy manufacturing or other areas?
Yeah, probably we can take this as an example. Yeah, for the future dairy manufacturing. Yeah.
I'm not. I guess my question would be, so you'll be looking for further support from the government in this. Is that your question? That you'll be seeking further government help in this?
Yes. Or maybe for any other JRCs. I mean, usually when the JRC funding sort of model finished, what could be the next steps from government's perspective? Yeah.
That's a very good question. I probably can't give you a definite answer right now, but I think it's an important area to consider. I guess with the JRC, the idea is to go from research into commercialization and business. So perhaps we're looking at more of in some ways, looking at commercial opportunities, business opportunities, which might relate more to trade. But from a research perspective, I'm not aware of anything particular at the moment, but it's a question I can take back to my my managers and see what thoughts there might be. But that's probably all I can tell you at the moment. But but there's still, I mean, within the JRCs, there's still, we're looking at future rounds. There'll be future opportunities for other areas. But as I said before, Australian government's looking to pursue science as an area for investment and engagement.
Okay, cool. Thanks, Evan.
I guess initiatives like the JRC sometimes it's more like a seed to get something like future collaboration, I think, and it may come in different form, maybe. Yeah.
Also, it also sets the foundations for develop networks and relationships in China, and to sort of progressing sort of give you the foundation.
That's
To give you the
Evan, sorry to
decide what you need to do next.
Sorry about this. You got a bit choppy towards just now. In the response.
Okay.
Okay. I think okay. Now, is there any more question from the audience? Yeah, Lynn. Yeah.
I think I probably just more like a comments rather than a question. During our CHIS Symposium in China, Australian Embassy really support us in this event, especially by the time Jonah introduced us to Wine Australia. So therefore we get a sponsorship from Wine Australia. We got 41 bottles of Australian wine and tasted along with those cheese. So what I'm trying to emphasize here is that under this scheme, we actually have a really good support from government, university, and also in order to collaborating with our industry partners and so on, carry on. Yeah, and we sincerely stand on that.
Okay.
Yeah.
I think that's good to hear.
Thank you.
All right, and I think thank you, Evan. We will move on to the next speaker. If you can stop sharing the screen.
Yep.
Thank you. Okay, and I would like to invite the next speaker. Now, we will now zoom in to really look at some of the exciting things that our students within the center have done. Okay, and our first speaker is Yang, and Yang. Let me give Yang now. Yang, can you share with us your screen first? And while you are doing that, from memory, I remember Yang, when we first started the center, Yang was still a very fresh PhD student, and he was asked to give a very important talk on behalf of his supervisor. And this time around, he'll give a talk for himself. Okay, so a brief background. Yang received his Bachelor of Environmental Science from Hunan University in 2013 and a master's degree in environmental engineering from the University of Science and Technology in China. He joined Monash University as a PhD student in November 2016 under the guidance of Professor Xiwang Zhang and Professor Cordelis Salamuya. His research interests focus on the synthesis of laminar membranes with 2D materials for desalination. Now, just in case what that all means, it really means that graphene, okay, as the title suggests. So, and on that note, Yang, please, yep, over to you. You are on mute. Okay, I'll just need you to.
Okay.
Okay. Thank you.
Okay. Yeah. So you can. So all of you can hear my voice now, right? Yeah. Okay. So today I would like to give my my talk with the title "Manipulating the Charge and Channel Properties of Graphene Oxide Memory for Desalination Purpose." The outline today includes the introduction, the research introduction, the games, and the aims, the research contents. I will give a short summary and the acknowledgement. Firstly, I would like to introduce the memory separation technologies. As we know, the memories has been widely used in different areas, such as the food, pharmaceutical industry, for desalination, for pure water production or wastewater treatment. Fundamentally, memory separation is according to the state-saving effect. Different memories, such as the microfiltration, nanofiltration, or reverse osmosis, the pore size is quite different from each other. And the separation is basically come from the charge and size exclusion effect. As the development of memory is highly relies on the materials development, graphene has already offered a alternative choice for the nanofiltration memory materials, as a graphene is a very thin materials, which is around 1 atom thickness, 2-dimensional structure with very high mechanic strength and chemical insertions. Compared to graphene, graphene oxide can be massively produced through the aqueous oxidation of the graphene graphite, and it can be processed in the visually the word processing, such as we can already use graphene oxide to develop the new kind of memory, which we describe as the laminar memory or 2D memories. Graphene oxide memory is very easy to get. We can use the pressure-driven or vacuum filtration, like we can use the deep coating or spine coating method to prepare this kind of laminar structure memory. As shown from here, the laminar memory has some unique properties, like it includes the nanosheets with some kind of like interlayer spacing. The water usually goes through this memory tutorial pathway, and the saving is come from the interlayer distance, something we describe as the size saving effect. But one thing like that has limited the dual memory separation is the swallowing properties of graphene oxide in water. As the dual memory is unstable in water, so it's not really practical for the industrial applications. One thing we can do is to stabilize the dual by cross-linking or chemical reduction or thermal reduction methods. For separation, we do want more than stability. So the research camp is shown here. Generally, after stabilize the dual memory, the channel size is become narrow, and some channels is blocked by the cross-linkers. So it generally come to the results that the memory shows lower water permeability, and we know precise separation is always the target for different application purpose. So we have to control the interlayer distance and the charge property of dual memory. Instead, the mechanism is very important to solve this problem. As we know, the dual memory, we want to develop the dual memory with high permeability and high selectivity. And we also want to know the transport mechanism that within the laminar structure. The things we can do is we can modify the existing nanochannels or by introducing new channels, such as we can make the memory like more porous. The first thing I have done here is I prepared the nanoporous graphene oxide. The second one, I want to turn the channel structure by a melt annealing method that enable memory to be more ordered. Thirdly, I prepare Janus memory with the asymmetric charge for desalination. The first thing is belongs to the new channels. The second one is the channel structure. The third one is the charge properties. So the first project is about the nanoporous graphene oxide. As we already know, the two-dimensional graphene lattice is non-permeable to any water or gas molecules. the thing we want to use it as a memory is by introducing the nanopores by the plasma or some kind of other method. We can create nano-sized pores with the precise diameter. If we want to use the laminar memory for desalination, we have to consider the channel size and the nanopore size as well. The memory development method is shown here. We firstly get the nanoporous graphene materials, and then through the filtration, we get a laminar memory. After that, we reduce the memory to get the nanoporous memory, which is somewhat reduced to stabilized. From TM or the SEM results, we get to know after introduction of nanopores, the letter size is around 1 micrometer, the pore diameter is around 3 nanometers, and memory around 20 nanometers. We compare the memory performance with those have not yet modified. We found actually the water permeance is significantly increased by 26 times, and solid radiation for the sodium sulfate is pretty similar. This result is very promising. When we go too deep about the mechanism, we found that actually the solid separation is dominated by the charge properties, which is come from the negative charge function groups on the implant jewel. Another thing is the size saving effect. The channel size when in water is around 1 nanometer, which is very close to the suffocate ions. So the mechanism of the nanopore introduction into the memory is basically we found that by layer stacking, the nanoporous laminar memory has additional transport pathway, and the charge and the self-saving effect enable the memory to be applicable for nanofiltration applications. So we know this memory is very potentially promising for the future nanofiltration to separate ions from water. Here is the summary for project 1. But we have already know the nanopores can actually introduce water permeability. We know the laminar structure is like composed with the nanosheets within the memory. So what's the relationship between the laminar structure and memory performance? We have already know from the lecture review, a more ordered laminar channels is beneficial for water permeance as well as the solid rejection. So one thing we can do is to prepare a more ordered laminar channels. From the previous study, we know corrugation happens during the solution phase reduction, which results in different laminar structures. And the melt annealing at 80 degrees is generally adapted for drying. However, it has been proved to be effective to produce reduced graphene oxide materials. So how about the melt annealing for thermal reduced archival memory preparation? Here is the method we developed for this memory. Generally, after filtration the memory on porous substrate, we reduce this memory in the oven. From the superficial view, we can see this memory after thermal reduction shows the similar trend as the high thermal temperature, which is the surface is become darker and more black. From some characterizations, we get to know after the thermal reduction, the channel is become narrower, and we see the change of function groups, and the reduced hydrophilicity and surface charge, indicating the melt annealing actually is able to reduce this memory. From the memory performance, we get to know the narrower interlayer spacing, leading to a reduced water permeance. However, we get to know the source separation is increased when we prolong the thermal treatment time, and the source selectivity between the sodium sulfate and some other ions is increased with time as well. So the melt annealing can enable tunable salt separation as well as a higher salt selectivity. One thing we want to know is to know the advantage of melt annealing compared to the high thermal annealing precise. So we prepare another memory at the same condition, except for the thermal temperature is much higher. And we compare these two memories with the same interlayer distance. We found actually these two memories compared with the high thermal treatment one, the melt annealing reduced the RGU memory shows both a higher word permeance and a higher selectivity. And we used the surface morphology analysis, and we found actually the high thermal treatment memory shows a more wrinkled structure with a higher roughness. So basically we attribute to the different separating mechanism to the more laminar channel that is stabilized in a melt reduction process. In such a more other laminar channels, the water go permeate this memory with a less resistance. So the memory shows a higher water flux, and the more other laminar channel also enable precisely the sieving effect towards hydraulic ions. So we achieve a higher selectivity. So this is the summary of the part 2. We get in the GO actually has various hydroxyl, epoxyl, and carboxyl functional groups, and the charge is very important in nanofiltration. So the third project we conduct is on the charge and separation relationships. From the lecture review, we already know that the charge dominant separation is very common in nanofiltrations for the reduced RGO memory. Generally, we got to know the salt separation follows the sequence of the salt rejection. Sodium sulfate is the highest salt rejection, but the magnetic chloride is the least separate salt ions when we use RGO memories. So the AB2 or A2B salt is quite different. By turning the memory surface from positive charge to negative charge, actually we get to know the salt separation is totally different. However, consider these two sides, we have no AB2 or A2B salt cannot be removed at the same time. One thing we get inspiration is from the Janus memory. Janus memory or the Janus conception is basically come from the Janus Scott with two phases. It's generally adapted for description of the asymmetricity, which may include the hydrophilicity or positive or negative charge. The structure can be A on B or B on A, or A from B or A and B. So it has two different layers. As graphene is generally is negative charge, if we introduce a positive charge layer, we can achieve a Janus structure. And the Janus structure actually have already offered us a further functionalization of GEO memory. From the memory preparation, we use the vacuum filtration, same as the previous two projects. After that, we adapt the spine coating method to modify the surface with the positive charge polymer, PEI. After surface modification by spinning coating, we found the memory is around 30 nanometers with a different positive surface charge and the negative layer. When we turn the charge properties by different polymer concentration, we actually found that with the increase of polymer concentration, the surface charge slowly change from like the top surface from negative charge to positive charge. Similarly, the bottom layer also slight change from the bottom negative charge to positive charge. Without the salt separation is generally gradually increased for the for the magnetic chloride. When the polymer concentration reached 0.1 gram per liter, we found both two salt ions can be removed at a higher rejection rate. So we found that actually we can use this Janus structure for manipulation of the memory performance. Similarly, we have done another thing is to increase the negative charge properties by increasing the geothickness. Actually, we found similar phenomena as the previous one with different separate mechanism. The proposed mechanism is shown here. By increasing the positive charge, actually we open the gates for the sodium ions, but close the gates for the magnetic ions. Different from this one, if we increase negative charge, we close the gate for the sodium ions, but open the gate for the magnetic ions. Basically, we attribute to this reason to the charge shattering and synergetic effect from both the top and the bottom layers of the Janus memory. The summary is basically come to this way, and when looking back these three projects, we have introduced like more channels by introduced nanoporous, and the secondly we turn the charge the Laminar structure by a melt reduction method, and thirdly we modify the surface with the positive polymer to create a Janus structure memory, which has a very high rejection for the both A2B and AB2 salts. That's basically the overall PhD study in MONASH University. Finally, I would like to give my acknowledgement to my supervisors and the scholarship I have received in MONASH. Thank you for your attention.
All right, Yang, thank you.
Yep.
Thank you for the presentation. Thank you for the presentation. And any questions from the audience for Yang? One, but perhaps the I just have a question on the maybe perhaps you can also answer this for the dairy industry.
Yep.
If they like to use this functional membrane that you have talked about, wonder what would be the direct application that people can try to explore to recover maybe waste water? Or?
Yes, we actually weight desalination is gaining attention due to the industry markets we have expect the cheese whey or cheese protein. Actually, desalination is always very difficult for the organic solutions like the whey or cheese products processing. One thing we can use the graphene memory is it has shown better unfolding properties compared to the polymer memory. Actually, we have already got reports from previous study, the unfolding or antibacterial property is much better since the graphene materials with antibacterial activities. At the same time, it should unfolding because it makes the memory more hydrophilic thus the organic fallen is not able to attach on the surface.
I see. And I think the like many things, like normally if sometimes this membrane may also need to be tailored according to a different application.
Yep.
Say if our industrial partner has interest to try out your membrane, okay, is there an avenue for them to bring a bottle of whey hot from the cheese factory, okay, and let you have a go? Is it
Yeah, it should be possible if we want to give it a try in the laboratory. Okay. But still, I think there are some other concerns may happen if we want the industry partners to use this kind of memory. Yeah, since it's a nanomaterial. Yeah.
Okay, thank you. Now, is there any question? I see Chen Yu is already here with the hands raised, eager to go. Now,
do you hear me?
Yes. Just before we start the next presentation, any more question for Yang? Any question for Yang?
I have one.
Oh, okay. So
I hands up because I have one question.
Okay.
So
yes.
You talk about annealing,
yep.
right? The mild annealing, and you compare the salt rejection ability of your film between mild annealing and high temperature annealing, and you also proposed a mechanism saying that one is less ordered, one is much better, highly ordered. And I just feel a bit curious, but obviously your presentation is pretty good. And one question is, because it's a film, I guess it's not very thick like rubber film, it's a film. So does the high temperature annealing might affect the mechanism strength of your film or any other like macro scale properties?
Yep. We have used the characterizations like the SPS or some other like the FTIR. We get to know the chemical combination is pretty similar, but the roughness shows quite different, and we have already got some hints from previous study. The laminar structure, when it is exposed to the thermal annealing, generally the structure become less ordered. Basically, we can draw this conclusion from the XRD. We see the peak is become less intense. The peak is less intense, and yeah, generally people say that it is because the laminar structure, or we see the lattice structure is become less ordered. And during this process, we compare the memory performance. The word permeance of the memory is higher. If we say this kind of memory with the same chemical combination, but with a higher permeance and selectivity, generally we attribute to the channel. The channel is ordered compared to the high thermal treatment. Yeah.
Thank you.
Yeah.
Okay.
I think we will now move on to the next speaker. Okay. Now, can I get you to stop the screen sharing, Yang, and then?
Yeah. I will stop sharing. Yeah.
Okay. All right, Chen, if you can share the screen, I will just give a very brief introduction for you. Everyone, Chen Yu obtained her, we normally call Matilda, yeah, obtained her bachelor in chemistry at Lanzhou University in 2011, and her master in chemistry at the University of Melbourne, 2016. Okay. Her previous work focused on ultrasonic modification of proteins and tuning the physical and functional properties of the synthesized organic liquid filled polymer microspheres. She started her PhD study at MONASH in May 2017 with Cordelia. She is currently working on microencapsulation, as you see from her title, to improve the bioavailability and the targeted release of bioactive ingredients. And this is really targeted at digestion. So in the future, she's aiming to focus on the design and formulation of functional foods with improved bioavailability and targeted release. So Chen-Yu, yep, over to you.
Okay. Can you hear me now?
Yes, we can hear you. Yep.
Okay. Okay.
Yep.
Hello, everyone. I'm Chen-Yu from MONASH University. Thanks for having me here to give this presentation. I'm in China now, so I'm not sure whether the internet connection is good or not. If any sound is chopping, just please tell me. Today, my topic is microencapsulation of curcumin by spray drying for improved bioavailability and targeted release. I would like to introduce some background information of curcumin first, which is the core material in this project. As food, curcumin is a well-known spice widely used in Asia cuisine. It is the main polyphenol of the curcuminoid class extracted from turmeric plant, and meanwhile can be used as coloring and antioxidant agent as food additives. Curcumin has numerous pharmacological functions, including anti-inflammatory, antioxidant, anti-tumor activities, etc. At present, the main challenge in the curcumin-related food is its extremely low water solubility, less than 11 nanogram per milliliter. The image shows below is the chemical structure of curcumin. Ketone formation is favored at acidic pH, whereas enol form is predominant at neutral and alkaline pH, which is easy to degrade. Microencapsulation has been widely used to improve the solubility and the stability of curcumin. However, most of these formulations were prepared in batch processes, which are difficult to scale up. Furthermore, the previous efforts focused on increasing solubility of curcumin. Little attention has been paid to control the release characteristics of the curcumin-loaded microparticles. So, to overcome these issues, whey protein isolates, WPI, were selected as raw material to fabricate curcumin WPI microparticles by spray drying. Curcumin is soluble in ethanol, up to 4 milligram per milliliter. Ethanol so was added as solvent for curcumin, and meanwhile as dissolvating agent for WPI in this case. Ethanol-induced dissolution was applied to promote the complexation between WPI and curcumin. Furthermore, calcium chloride was also used, leading to calcium ion-induced protein cross-linking to achieve sustained release of curcumin in the small intestine circumstance. The image shows the solution appearance when 25 milligram curcumin dispersed into 200 milliliter different solvents. Curcumin is nearly water insoluble. With the addition of WPI, the solubility of curcumin increased to 19 microgram per milliliter. This is because the main component of WPI is lactoglobulin, a type of lipid cleanse, which enables to bind and transport hydrophobic small molecules like curcumin. On the other hand, curcumin is soluble in organic solvents like ethanol. Therefore, the more ethanol introduced, the higher solubility of curcumin measured. UV-vis absorption spectroscopy was applied to study possible interactions between dissolvated WPI and curcumin. A peak at 273 nanometer is characteristic of the aromatic amino acids in proteins. It was blue shifted to 268 nanometer with the addition of curcumin. This indicates a change in the microenvironment around the WPI due to the interactions between curcumin and the dissolvated WPI. The fluorescence spectra of 5% WPI with varying concentrations of curcumin in pure water and 30% ethanol systems are shown in this slide. After all calculations, briefly, the dynamic quenching rate constant, the KQ value, which is highlighted in yellow circle in the table, was much higher than the coalition quenching constant 2 × 10⁻10 in both two systems. It recommends the major role of static quenching in the formation of curcumin WPI complexes, no matter with or without ethanol. Additionally, the thermal quenching constant, KSV values highlighted in the blue circles were proportional to temperature in the 0% ethanol system. On the contrary, the KSV values were inversely related to temperature in the 30% ethanol system. These observations suggest that ethanol-induced dissolution promotes the formation of curcumin WPI complexes. After spray drying, curcumin WPI complex microparticles with various compositions were prepared. Generally, the microparticles shows spherical, wrinkled, and pot-like shape. The morphology of microparticles became more wrinkled once ethanol and calcium ions were introduced. This slide summarizes the parameters and compositions of sample 1 to 6 as prepared. I would like to emphasize 2 points. Firstly, in the spray-dried state, the all curcumin contents of sample 2 to 6 upon dissolution, which is highlighted in yellow circle, or up to 3.47 milligram per gram particle, or mostly 1 order of magnitude higher than that of sample 1 without dissolution. Secondly, all samples displayed good encapsulation efficiencies, higher than 87%. As I mentioned before, curcumin is unstable at neutral and physiological pH. Its degradation products include ferulic acid, annealing, etc. This stability test showed here shows that free curcumin was degraded by over 60% within 30 minutes at neutral and alkaline pH. By comparison, more than 70% of the curcumin were stable in the curcumin WPI complex in the PBS buffer, SGF, and SIF after 3 hours. This reveals that the generation of curcumin WPI complexes effectively enhanced the stability of curcumin. This slide shows the release characteristics of curcumin from curcumin WPI complex microparticles in the simulated GI tract. In Figure 1, initial buster release of curcumin was observed in all samples, caused by the surface distribution of curcumin in matrix-type microparticles. Ethanol-induced desolvation improved the digestibility of WPI, leading to a rapid release of curcumin observed in sample 2 and 3. Cross-linking at higher calcium ion concentrations brought it back to be gastro-resistant, exhibiting sustained and intestine-targeted release characteristics observed in sample 4 to 6. The disappearance percentage of curcumin with the first hour intestine incubation in sample 2 was more than 20%. It was much higher than its degradation result in the previous stability test on the same condition, which was about 10%. To investigate these differences, 0.05% twin 80 as an emulsifier to fully dissolve curcumin was added into the simulated GI tract. In Figure B, we can see that their overall release trends were similar with those without twin 80, and the corresponding curcumin contents detected were comparatively larger. This presumably due to the dissociation of curcumin WPI complexes with the enzymatic hydrolysis as a function of time and the resulting formation of free curcumin released. I have to mention that I'm on a research exchange in Suzhou University, China at this moment. I incorporate these complex microparticles into yogurt and investigate the release characteristics of curcumin with a very new near real dynamic in vitro human stomach system. By using this human stomach system with a similar stomach morphology, dimension, and wrinkled inner structure to those present in human, I could look into the crucial micro-scale processes in food digestion and neutral release, such as gastro-sieving, gastro-emptying, and the pH variation of digestion. This new human stomach model is a very interesting part, and the data is still it's not. Hopefully, I can present the data next time. And last one is the antioxidant activity of raw WPI and the spray dry. Oh, last one is the antioxidant activity test. The antioxidant activity of raw WPI and the spray dry WPI curcumin complex microparticles were examined. Figure A demonstrates that the curcumin WPI complex microparticles had higher antioxidant activity than raw WPI alone. A comparison of Figure A and Figure B reveals that when entering the small intestine and getting hydrolyzed, raw WPI and the complex samples possess similar antioxidant activity, meanwhile being much larger than those without enzymatic hydrolysis. Compared to Figure B, Figure C indicates that curcumin degradation resulting from the effect of incubation time has a minor influence on their antioxidant activity. To draw a conclusion, first, upon dissolution, the maximum oral curcumin contents loaded was up to around 3.47 milligram per gram particle, approximately 1 order of magnitude higher than the sample without dissolution. Secondly, the spray-dried complex microparticles possess the better solubility and the stability at physiological conditions compared to curcumin alone, enhancing the oral bioavailability. The properties of curcumin was not oxidized after spray drying and retained in the form of complex microparticles. Fourthly, the complex microparticles dissolved and cross-linked at 3 to 10 millimole calcium chlorides displayed higher resistance to the GI tract. That's all. Thank you for your attention, and thanks my supervisor, Professor Cordelia Salomia, Professor Victoria Heritus, Professor Meng Wai Wan, and also Dr. Yong Wan, and all the friends and the members of ACGRC. Thank you.
All right. Thank you, Matilda. Thank you for the presentation. Now I'd like to invite questions from the audience. If there's any question for Matilda. I think something I'd like to perhaps add this matrix that you described. Okay, if people are interested to use it for other materials, I believe curcumin is mainly a model material. I think it is also applicable to other hard to dissolve active ingredient, I think. Would you like to comment?
Yes, because the main component of WPI is lactoglobulin, which belongs to a family of lipoclains. It enables to bind and transport hydrophobic small molecules. So any other, not any, but many other small molecules which is hydrophobic, I think can be applied in this system.
Yes, and I would like to encourage everyone, anyone who has any active ingredient, particularly hard to dissolve in water type of ingredient, to also, if you like to encapsulate them as part of your product development, you can certainly contact Matilda, to have a go. Yeah.
And also just want to quickly comment, Matilda mentioned about the system that she's currently using at Suzhou. She's still in Suzhou at the moment, or actually maybe still in her hometown, but she's going to restart soon. If you have a copy of the newsletter that we sent out with the invitation, you can see a picture of that digestive model that she mentioned. Matilda, just want to have a quick question, because I think I was wanting to ask you before. You mentioned that you put your microparticles into yogurt, right?
Yes.
For your new, have you actually tried and see whether it's going to affect the texture of the yogurt, or even whether that's going to affect the grittiness or even the taste as well, because you put curcumin there?
Actually, I carried out a sensory test. So I asked 40 students to try and test my curcumin yogurt, and based on the smell, the smoothness, the creaminess, and other standards, yeah, they like it. It's pretty good, and they actually the curcumin is almost non-smell, but it didn't change the structure or the texture of yogurt very much.
Thank you.
But the yogurt is a bit yellow. It's a little bit yellow, but it's okay. It didn't affect the smell.
It's a very rich milk, something. Okay.
Thank you.
All right, and I think that we have come to the we have one message to everyone. Our newsletter. Okay, yeah, that's from just more information on newsletter. Now we have come to the end of the first session, everyone, and as I mentioned, I'd like to once again thank all the speakers for the great effort. Yeah, in this new format for this this session. And what we're gonna do is we'll have a 5 minutes break, and we'll resume the symposium. The next session will start at 1. My clock here is 1:13, so we'll start at 1. Oh, New Zealand time. Sorry, we'll start at 11:20. We will resume at 11:20, the next session. And normally now is the time where we normally invite you to have a tea or a muffin, but we are all virtual now. So I'll still do the same invitation. What I'll do is I will share my screen, and I'll just show you a picture of some bread that just to whet your appetite. Okay. Now, everyone, when Lynn was talking about increase in baking, I'm guilty of that during the break. You know, you have nothing to do, DIY, and just to share some of the sourdough and some other baking that I've done.
Hi, Mongwei, are you playing guitar or playing music? We can't see you.
Just having a practice.
Yeah, I think it's 11:20 now. So we are going to start next session.
Okay. Oh, back to you.
Yeah. First, my name is Yong Wang. I'm very glad to join this ACJRC Symposium because I have working with 3 organizations who's all involved in the JRC. First, I worked with Coffee Nutrition Health Research Institute in Beijing between 2011 to 2019. And I was the main contact for the ACJRC from the Coffee now. Last year, I joined MONASH with Department of Chemical Engineering, working as a postdoc. And this year, this February, I joined USW as a senior postdoc here. And my research here with USW will focus on 2 areas. First is food rheology. In particular, we want to develop the rheological testing method, which can connect it to the food processing and sensory perception. The second area is the interaction of food ingredient in colloid, and the potential application will be for the emulsion stabilization and improve the encapsulation efficiency for microcapsules. And our first speaker in this session will be Naomi Whedon. Naomi, can you start to share your screen now, and I'll do a brief introduction. Naomi is the PhD student with the University of Queensland. She has been working on the encapsulation of probiotic for extended shelf life in ambient temperature, very challenging topic. And before joining UQ, Naomi has owned her own business, a startup on chocolate. So I'm very interested to see how she will combine the research capability, also her industrial experience to solve this technical challenge. Now is your time, Naomi. Please go ahead.
Hello. I'm doing my research on improving thermal stability on probiotics in mostly liquid dairy foods such as yogurt and milk. So here we go. My supervisors are Professor Besh Bandari, Dr. Lai Tran, and Dr. Young. So, move the slide. I apologize for this. So I'll first give you a bit of background on my research. So probiotics are defined by the World Health Organization as live microorganisms which, when administered in adequate amounts, confer a health benefit to the host. The key word in this definition to me is that they have to be live. So to give that benefit, they must be alive. Probiotics are present in many foods. They are either naturally or they have been purposefully added. However, the range of foods currently available is fairly limited. This is due to the fact that they are quite sensitive to many stresses that foods go through when they are being processed, such as oxygen levels, high or low pH levels, shear stress due to mixing, and most importantly for me is thermal treatment, so high temperatures. And this is where my research comes in. So microencapsulation can be used to protect the probiotics from a lot of these stresses, especially temperature. So the overall objective is, in my research, is to develop a microencapsulation matrix which will encapsulate the probiotic bacteria to provide protection during pasteurized thermal processing in a liquid food, mostly liquid foods such as yogurt and milk. So these microcapsules would be incorporated into the food product and prior to pasteurization, with the aim that after pasteurization, the food will actually still have viable probiotics in them, but actually be safe to eat, as all other bacteria and nasty pathogens have been killed during the thermal processing. Furthermore, they could also possibly be shelf stable, whereas currently most probiotic food products are refrigerated products. So this is a brief overview of my methodology. I'm actually working with 2 probiotics at the moment, Lactobacillus and Rhamnosus GG and Bifidobacterium animalis subspecies lactis. I'm going to call them LGG and BB12. These are then incorporated into my matrix, so they're encapsulated, and then these microcapsules are then incorporated into food. They are then thermally treated, and then the viable probiotics are measured, and storage trials and measuring the different parameters of the food, changes to the food such as pH, the size of the microcapsules, all of these sorts of things are also measured. These are taking place at both refrigeration temperature and also at room temperature. Okay. Okay, so LGG and BB12, they've been successfully encapsulated within the internal water phase of a double emulsion, which is the basis of my encapsulation system. In this image here, there we go. You can actually see some BB12 in the internal water phase. They're wriggling about. Encapsulation efficiency for BB12 is actually over 95%, and it's 93% for LGG, so that's pretty successful. And when subjected to heat treatments such as 75 degrees centigrade for there are over 10 to the 7 CFU per gram of LGG and just under 10 to the 6 CFU per gram for BB12, still viable. So that was pretty good news. So once this is done, this double emulsion is added into milk and yogurt. I was actually pleasantly surprised that it remained stable, as double emulsions are notoriously unstable. Unfortunately, it destabilized once it went through the modified UHT plant at 75 degrees for 30 seconds. It completely destabilized, and that was not that unexpected, to be honest. This actually brought me to my next stage, however, which is incorporating a gelling mechanism into the secondary water phase. So the hydrocolloid of choice was alginate. It's very well recognized. It's safe to use, food grade safe, and it's recognized as a good encapsulator for probiotics, as well as having, it's also very easy to use, as you just cross-link it with calcium chloride and it gels beautifully. So initially I made some macro beads. This was produced using the extrusion technique. I used different concentrations of alginate, 1%, 1.5% and 2%. The 1% was deemed not good enough, so we stuck with the 1.5% and the 2%. Once we determined that they could be gelled, we then transferred the technique to the impinging aerosol encapsulating technique. This is this machine here. So this is where your matrix containing your probiotics are atomized, and in the opposite direction you have the cross-linking agent, which is calcium chloride in this case, atomized at the same time. And so the alginate gels, these are then collected at the base and then filtered, and I get these little tiny microcapsules here. These are then incorporated into my liquid food, which is yogurt and milk. This here is this is just a light microscope image. It's a 5% alginate, this one, within the secondary water phase, and it's at time. There we go, next slide. So these were then incorporated into milk and yogurt. These are the shelf life storage trials for milk at both 4 degrees and room temperature. There wasn't that much difference between the temperatures, slightly higher in both cases for room temperature. These are only preliminary storage trials, and these are for the 1.5% alginate. So, another important aspect is the microcapsule size. This is an important problem because if it is too large, there are some major sensory impacts. Nobody wants to eat yogurt or milk that has a gritty, sandy texture. However, literature searches indicate that there is a high correlation with a larger microcapsule providing greater protection against environmental stresses such as thermal treatment. So we actually want a nice balance, a microcapsule that is large enough to provide enough protection, but small enough that you can't tell it's there. So we found that storage had no significant impact over the microcapsule size, neither did the storage temperature. However, the food did have a significant impact on the microcapsule size. Milk, which is at a neutral pH, the microcapsule was at approximately 39 microns in size, whereas in yogurt it was 18 microns in size. This would be due to the pH difference. So alginate has a habit of swelling in neutral pHs, and as a result, we have the size difference. It was actually significantly different. Now, I wanted a slightly larger capsule than 18 microns for the yogurt, and so I have been adjusting the encapsulation process, and I have currently managed to get it up to 25 microns in size. So this was quite good, and I believe it will actually improve the thermal protection for the microcapsules. So we actually going to come into the most important stage now, which is the viability after thermal treatment. They are definitely alive after thermal treatment with my microcapsule. So to determine whether viability, I use confocal laser scanning microscopy using the Backlight Live Dead kit. This has 2 fluorescent dyes, Cyto 9, which is the green. So you can see in these images here, you see little green bacteria, and that means they are alive, and propydium iodide, which fluoresces red, and that binds to them when they are dead. So red is dead, green is alive. So this image here, this is untreated milk with LGG. You can see this is a microcapsule. You can see there's quite a lot of green bacteria here. This is actually treated. So there are some green bacteria here. This is actually for the 1.5% alginate for the milk. On this side here, we have treated yogurt. So in this here, I have live bacteria, and in here I've got a couple of dead bacteria. So these are both treated. This here is the microcapsule within the yogurt, and this is actually, so you can see the fluorescence, I took out the bright light at the back. So they are definitely alive. They are not growing on agar at the moment. I'm having some issues with that. So what I'm planning on to get counts, I'm planning on using Z stacking, using the CLSM, and using software such as ImageJ to actually do some counts. I have had some success with this at the moment. Just in this last week, I've managed to get some counts, but I need to work out some density issues with the how many capsules are underneath the microscope. So to get actual proper numbers for the yogurt and the milk. So this was great news with my samples that they are actually working, and this isn't actually the final microcapsule. So this is the 2%, but I am actually investigating changing the internal water phase to improve viability. So furthermore, I can also find movement when looking at a light microscope. And after the heat treatment, you look down the microscope and you see, like that first video I showed you, little bacteria wriggling about within the microcapsules, which was great. So from here on, in conclusion, I currently have live bacteria, probiotic bacteria, present in the microcapsules in the yogurt after thermal processing. This was my aim. I found that storage temperature and time has no impact on capsule size, and after 4 weeks, there are still live bacteria using the CLSM in the microcapsules. I'm continuing to improve the encapsulation process. So to improve both stability of the microcapsules and to improve the viability. I want to investigate the morphology of the microcapsules using SEM and ESEM, and to further investigate an additional matrix components for greater thermal protection, which I have just initially started. Hopefully the ImageJ software and Z stacking using CLSM will allow me to get some actual numbers for the heat treated yogurt and milk samples, and I'm hoping this is so far is looking fantastic. And I have yogurt that is has probiotic bacteria in it after thermal treatment. So this is what I'm aiming for, and so far so good. Thank you very much for listening to my presentation. I'd like to acknowledge everybody at the Australia-China Joint Research Centre, and especially my supervisors, Besh, Lai and Yong, and also an undergrad student that was working with me as well. She helped me quite a bit as well. So thank you very much. And that's it. Any questions so far?
Thank you, Naomi. Very interesting story. I see there are 2 questions from Sophia. Can you see the chat? Or maybe Sophia, can you repeat the question? Because I think the first one is pretty short.
Yeah, hi, this is Sophie. I actually have 3 questions, but so the first one is, can you possibly share the dose density for the beads, like 10 to the 9th or 10 to the 8th or something like that? And then have you taken plate counts to identify the probiotics, whether they're live or not? And actually actually the third one would be, have you done shelf live test? But I assume you probably haven't done that yet.
I've just done some preliminary shelf life tests at the moment, just 4 weeks at the moment. I want to get everything absolutely stable with my microcapsule, and then start them 10, 12 weeks of or more of shelf life testing. I do have numbers for the untreated probiotic yogurts and milks. They are currently at 10 to the 8, 10 to the 9 for per gram of milk or yogurt. So what were the for the treated, the probiotics, I believe, are quite stressed after I've been messing about with them. And my unencapsulation process is also quite stressful to them. And so I am having a few issues with plate counts. My agar is very specific for those 2 probiotics to contamination. So yes. Does that answer the question?
Right. So 10 to the 9th or 8th, would that be for the beads or actually in yogurt?
No, that's the yogurt.
Oh, that's a lot.
Yeah, I know.
So what's the density like for the beads?
Pardon? What's the?
So you have initial beads, right? So
Yes.
for the beads, what's the density like?
I have to measure that at the moment. We've had a few lab shutdowns for some pieces of equipment, so I haven't been able to actually measure that at the moment.
Okay, but so like how much beads have you taken in each yogurt? Like, for example,
sorry. Now I understand. It's between 1 to 2% dosage. So,
okay, so it seems like if you have 10 to the 8th in the yogurt, then the beads would be 10 to the 10th or something like that, right?
yes, that is approximately right. Yes, 10 to the.
Okay.
The initial is approximately 10 to the 10.
Great. Okay. So after heat treatment, did you say you haven't done any plate counts results yet, right?
No, I haven't got that yet. No.
Okay. Right. Thanks.
Okay.
Okay, great. Our next question is from Pooja. Nami, can you see the question?
No, I can't get the chat up. Hang on a sec.
Yeah, I'll read the question for you. Was there any further effect on microencapsulation with the different variety of yogurt and milk, like whole fat, skim milk, etc.? Or are probiotics used only for a specific kind of milk or yogurt?
So what type of milk did I use and what type of yogurt?
Yeah, and do you think it's specially designed for this kind of milk, or it can be used in all kinds of milk and yogurt? That's the question.
Okay, so I believe it could be used. I think the encapsulation process has to be possibly developed for both yogurt separately to the milk. So I'll find the slide. Here we go. So I atomized the microcapsules at, we call it 3 bar. So, and that received, so this is actually the same microcapsule here. Same set of microcapsules that have just been split and put into milk and into yogurt. And these are the changes in the microcapsule size. So I believe possibly adjustments would need to be made for, say, yogurt. So slightly larger microcapsule would be available in the yogurt. So the pressure, I used 2.5 bar instead of 3 bar. And that made a slightly larger microcapsule. So there might be slight differences in the encapsulation process, as the milk has a higher pH and it causes the expansion, the swelling of the microcapsule as a result. So if I haven't measured it yet, but the microcapsules that went into the yogurt, they would possibly maybe be up to 50 microns in the milk. I need to actually measure that though to be certain. So there may be slight differences there, but that's the only difference so far that I have discovered. Okay.
Okay. Our next question is from Amish. Amish, I have unmuted you. Do you want to repeat the question?
Yeah, Naomi, I think you already answered this. Nice presentation. Good question answer probiotic. But the question was around probiotic CFU per gram in the yogurt. I think you mentioned 10 to the 8, 10 to the 9 at the start of shelf life. Is that correct?
Sorry, it's very quiet, I find.
Yeah.
Sorry, could you repeat? I couldn't hear the question very easily.
Sorry, can you hear me now?
All right, Amish, I think your microphone is a bit low. I will repeat the question. He asked, what level of probiotics did you achieve in the yogurt, like in CFU per gram?
Oh, initially? The initial stock, is that the question?
Yeah, I think it's the initial and how much of them can survive. Probably both of them, if you.
After encapsulation, after treatment, I'm not entirely certain yet. However, after encapsulation, I know that majority of them do survive. The encapsulation efficiency is 95% for BB12 and 93% for LGG. So when I have my stock solution is approximately 10 to the 10. That's in water phase 1. And so in the microcapsule, it's approximately 10 to the 8, 10 to the 9, if that helps.
Yeah, I think you have answered the question.
Oh, okay.
And our next, probably the last question is from Simo. Simo, I have unmuted you. Do you want to repeat the question?
Yep. Hi, Naomi. I'm just wondering if you're planning on doing any further research about the to analyze the survival of the bacteria through the GI tract in the yogurt matrix.
Yes.
After.
Yes. What I actually didn't mention is I'm planning on doing full release studies. I also would like to do sensory if possible. So that is my next plan. So viral and basically through the GIT tract to see how they survive. I want to make certain that they actually are released in the correct place, as I want to give the human the health benefit, not the sewage treatment plant. So yeah.
Yeah. Cool. Thank you.
Thanks.
Okay, I see another question pop up from Christoph. Chris, do you want to repeat the question?
Yeah, sure. Thanks, Naomi, for your presentation. My question is also very similar to the others. Did you measure the percentage of survival of your probiotics as a function of time?
Sorry, what was that?
Did you measure the survival rates of your probiotics during the function? As a function of time.
For my preliminary storage trials, do you mean?
Yes.
Basically, I just measured them every single week. So it was weekly. I took samples out and I measured the viability using
Okay.
plates. Yeah.
Okay. Thank you.
Okay, I think we have this last question and from Maxim. And do you want to repeat again? After this question, we'll go on to the next speaker.
Yes, just a quick question about what if the product was homogenized? Like, do you know what the homogenization process would do once the capsules are in the milk?
Actually, no, I don't. That's a good question. So far, I've always added to a pre-homogenized product. So, that's a very good question. I do use an Ultra-Turex to actually add the microcapsules to the food, the yogurt or the milk. But actually, I think I should probably test that. Thank you. So that might have a big impact. Yeah.
Okay, thank you, Naomi. Very interesting, and you see a lot of questions. Our next speaker will be Shi Yuan. Shi, would you share your screen while I introduce you? And Naomi, please. You have stopped sharing. Shi is the PhD student with MONASH University since 2017. Before joining MONASH, she got a bachelor degree in applied chemistry in 2011 and a master degree in environmental engineering at Tsinghua University in China in 2014. He focused on the development of novel advanced oxidation process for water and water treatment. And in the ACJRC project, he focused on the fabrication of high-performance nanocomposite membranes with anti-folding ability. And Shi, you can start your talk.
Okay, thank you for your introduction. And I'm Shi Yuan. Thanks for having me here to share my work on membrane technology. Different from Yang's work, my research is to minimize the nano-wrinkles of reduced graphene oxide membrane for the rejection of small cations like sodium chloride. Firstly, let me introduce membrane to you, to some of you may not familiar with. Membrane is a film which can separate some target components from a mixture, and membrane technologies have been widely used for seawater desalination, gas separation, wastewater treatment, as well as dairy and food manufacturing. However, conventional polymeric membranes are normally suffering a trade-off between permeability and selectivity. And at the same time, biofouling and bacterial fouling is another problem, which leads to which requires frequent washing of membranes, and of course increases the running costs. So new material-based membranes are highly required. And as the emergence of graphene oxide, graphene-based two-dimensional materials have drawn a lot of attention, particularly graphene oxide, as mentioned by Yang before, can be easily fabricated in aqueous reaction and has been attempted to prepare high-performance membranes. Graphene oxide nanosheets contain abundant oxygen functional groups, and they can be feasibly assembled into membrane. And by vacuum filtration, spin coating, or solvent evaporation, these graphene oxide membranes exhibit unique 2D structures, which allows fast water transport as well as precise molecular sieving. And due to the good hydrophilicity of GO-based membranes, also show a good anti-fouling ability. However, when we use geomembranes in water, the hydration of oxygen functional groups will lead to the significant swelling of GO. And the interlace space, which is the size of the 2D nanochannel, will increase from sub-nanometer to several nanometers, which will lead to the decrease in their selectivity and stability. And if we apply geomembrane in water for a long time, it will even redisperse into water. There are several strategies to overcome this problem, and the most straightforward way is to remove the oxygen functional groups. And it has been reported that the functional groups could be partially removed, and the interlace space can be adjusted. And this RGO membranes has shown high stability and selectivity, which is promising for saving small molecules or ions. However, we are also facing some challenges, and like this one, like researchers prepare RGO membranes with chemical and small reduction methods. Even the chain size decreased to 4 angstrom, which is quite small, way smaller than the hydrated sodium cation. But the rejection to this cation is quite low. It's way low than the theoretical value. Only expect 40% of rejection of sodium chloride. So this phenomenon makes me think there might be some other channels for water and salt transport other than the well understood 2D nanochannel. So the understanding of other channel is the objective in this work. So this is how I prepared my membranes. I prepare different RGO membranes with three reduction methods: thermal reduction in air, thermal reduction in vacuum, and HI vapor reduction. The resultant membranes are named as air RGO, vacuum RGO, and HI RGO. Ideally, these RGO membranes will exhibit different structure and chemical properties. Firstly, I investigate the structure, the cross-sectional SEM images of GO and ARGO. We can observe the decrease in their thickness. And we also investigate the reduction degree by XPS and the interspacing by XID. By comparing the oxygen to carbon atomic ratio, ARGO shows lowest reduction degree and largest interspacing, which means thermal reduction in air is a moderate reduction process comparing with the counterparts. Then we try to identify the structure characteristics. Firstly, the 2D nanochannel S, which is well understood before by other researchers. And we can find that all the size of the effective size of this 2D channel are similar or smaller than the hydrated sodium chloride. So ideally, these three membranes can effectively reject sodium chloride. And for vacuum RGO and HI RGO, it's even the channel size is even smaller than the water molecule. So ideally or theoretically, these two RGO membranes cannot allow water to go through. So let's see the performance we conduct. So we use investigate their performance in a pressure-driven process with 1 gram per liter sodium chloride solution. As illustrated by this figure, RGO membranes with smaller interspacing exhibit smaller water flux. However, low sodium chloride rejection is also observed. By contrast, LRGO with larger interspace shows effective sodium chloride rejection. So this phenomenon further indicates that there must be some other channels. So we then double check the order of 2D nanochannel as mentioned by Yang before. The order of the channel is a key factor when we considering the rejection of small ions. So we analyze the RGO to GO ratio in thickness and in the interspacing. If this two ratio matches well with each other, it means this membrane is uniform. And we can only, the table shows that we can only find ARGO shows similar ratio in both thickness and interspacing. And while for other two reduced GO membranes, the structure is quite disordered. So this kind of structure disorder will lead to the formation of nano wrinkles, which is the factor we think which is very important. So we then directly characterize the nano wrinkles by SEM and FM, and we can easily observe the difference in their surface structure and the density of the nano wrinkle. We then further analyze the FM results and got the surface roughness and surface area, which are the indicators of the nano wrinkle size and the density respectively. And we also developed a model to calculate the wrinkle volume. And we can see from this figure, the LRGO shows the smallest wrinkle volume. And by comparing the wrinkle volume with the sodium chloride permeation rate, we confirm that the salt will only transfer in the non-wrinkle freely, while water can go through membranes in 2D nanochannels like in here, while when the interspace is large enough. So we propose a new mass transport mechanism in RGOMs. There are two types of channels in RGOMs, the 2D nanochannel and the nanowrinkles. When the interspacing is too small, like that in vacuum RGO and HI RGO, water and salts can only go through the nanowrinkles without any selectivity. So these membranes will show smaller water flux and lower sodium chloride rejection. When the size of 2D nanochannel is between water and the hydrate sodium, small amount of salts will go through the nanowrinkles, and the water can transport in both regions. So the sodium chloride rejection rate is enhanced. In conclusion, nanowrinkles in RGOMs serves a non-selective channel for water and salt transport, and nanowrinkles have more significant impact in salt rejection of RGOMs rather than the 2D nanochannels. And LRGO membranes exhibit enhanced sodium chloride rejection and relative high water permeance, benefiting from their minimized non-selective nanowrinkles and slightly narrowed 2D nanochannels. Last, I would like to thank my supervisors and Professor Zhang and Professor Mona and the group members from ACGRC and the MC Lab and scholarships. Thank you very much.
Thank you, Shi. Let's see if anyone have question. You can unmute yourself or you can raise hand, let me know.
Okay.
Hello. Hello, Shi.
Hi.
Yeah, I have one question for you. So nanowrinkles can actually be the water transport channels without any selectivity. So is there any other kind of unselective nanochannels except for the nanowrinkles you mentioned here?
Yes, indeed, there are some other non-selective channels like defects and yeah, because but in my system, the membrane is thick enough to prohibit the formation of the defects. So this is not a factor in my case. Yeah, but there are some other channels. Yeah, if the membrane is too thin.
Yeah, I mean, if the nanowrinkles can be tuned in order to achieve the targeted performance, so some other nanochannels like you mentioned device maybe also be beneficial if we can achieve controllable modification approaches like through the geometric design or something. Yeah.
Yeah, might be, but my target here is the rejection of small cations. So when it goes to sub-nanometer, it's very difficult to turn the defects or turn the size of the defects. So it's quite challenging, but it's a way to further increase the performance as well. Yeah, thank you.
Yeah. So is that possible to insert some quantum dots into the nanochannels to block the unselectivity?
Yes, it's a choice. It's very yeah. It might work. Yeah.
Okay. Yeah. Thank you.
Thank you.
And I think we have another question from Chris Bunning. Chris, do you want to repeat your question?
Yeah, sure. It's just a question. How does the flux on the geomembranes compare to competitive technologies which are in the industry already, such as ceramic membranes or polymer membranes?
Thank you, Chris, for your question. I would say, yeah, geomembranes. The water flux of geomembranes is quite different. I mean, after the reduction, the water flux is quite low and much lower than the commercial ceramic membranes and polymer membranes. But since my membrane is aimed to reject small items, so it's reasonable low. But some of the geomembranes has larger water flux, way larger than the ceramic membrane and polymer membrane after some certain modification, like the cross-linking by organic compounds. But it depends on the application. Like if you want to apply the geomembranes for UF or nanofiltration or ultrafiltration, so different modification can be used to achieve both high water flux and high rejection of your target products, target molecules.
Okay, is there any further question? If no, we are going to move to the next speaker. Thank you, Shi. Thank you very much for your presentation. And